Neurological Disorders Lesson 2.2 Differentiated. How do our neurons signal electrically? This is one of two lessons that introduces the action potential. This lesson, 2.2 Differentiated, contains many more details than the other lesson, lesson 2.2. And this lesson is best for classes where students have already mastered concepts of diffusion, threshold, and the impermeability of the cell membrane. If your students need to review these concepts, use lesson 2.2 as it reviews those concepts as it presents the action potential. Additionally, in order to allow for adequate time for your students to complete the activity, you may want to allow 2 class periods for this lesson. The goals of this lesson are to introduce students to the process by which our neurons signal electrically, the action potential, and to explore the consequences of disrupting the signal in the action potential. At the end of this lesson, students should be able to describe how sodium and potassium ions flow in and out of the axon to create the action potential, describe how the action potential moves down the length of the axon, and describe how Novocaine works. We'll achieve these goals through a Socratic discussion and modeling the action potential. To prepare for this lesson, you'll need to review the key scientific concepts that we presented in it. And a word of caution, students find the scientific concepts presented in this lesson to be challenging, so it is imperative that you take some additional time to ensure that you understand the concepts to help guide your students. Those concepts are the types, locations, and relationships between different neuronal signals, that dendrites generate electrical input signals, that axons generate electrical signals, and that presynaptic terminals generate chemical signals. We'll also discuss the creation and conduction of the action potential, the axon's electrical signal. This involves several key scientific concepts, including electricity, the impermeability of the cell membrane, ion channels, and the membrane potential. The lesson also discusses the flow of sodium and potassium ions during the different stages of the action potential, including getting to threshold, depolarization, and hyperpolarization. You can review this scientific content in the background reading provided to you in the teacher primer, the teacher manual, and the student workbook. The teacher primer provides in-depth knowledge about the scientific content presented in the lesson. The teacher manual or lesson plan provides a minute-by-minute explanation of the lesson structure, including instructions on how to manage the discussion and activity. And the student workbook provides additional explanation for students. You'll need to be sure to print the activity worksheets for your students. And before the lesson begins, you'll need to prepare action potential model setups. You'll need one setup for each group of around 3 to 4 students, and it's easiest if you package each in a Ziploc bag to save time during class. Each action potential model setup includes approximately 100 black-eyed peas, approximately 100 lima beans, 3 blue toothpicks or pipe cleaners, 3 red toothpicks or pipe cleaners, and 3 black toothpicks or pipe cleaners. The key point of the lessons due now is to engage students with the concept of neuronal signaling. We'll get there by brainstorming how Novocaine works. After allowing students time to brainstorm and share their ideas, you'll animate slide 2 to show students that Novocaine stops our neurons from signaling electrically, which will lead directly into our discussion. The key points of the discussion are to introduce the types of neuronal signals, including the electrical signal down the axon and the chemical signal across the synapse. The discussion also presents the creation and conduction of the action potential, which depends on the following key scientific concepts, which will also be discussed: electricity, impermeability of the cell membrane, ion channels, and membrane potential. The discussion also covers the flow of ions during the stages of the action potential. Those stages are getting to threshold, depolarization, and hyperpolarization. We'll get to all of these key points with a Socratic discussion. The discussion starts with an introduction of the types of signals neurons send. Neurons signal chemically via synapses and electrically via axon. This lesson focuses on how neurons send electrical signals down their axons, and we'll focus on how neurons send chemical signals across synapses in Unit 3. You'll use slide 4 to introduce students to the action potential, which is how neurons send signals electrically down their axons, and they do this using the potential energy stored in their membrane potentials. The discussion then elaborates on both concepts of electricity and membrane potentials. First, looking at electricity, the flow of charged particles or ions on slide number 5. Since ions can't flow into and out of the axon, this leads into a discussion of the axon's ion channels and pumps on slides 6 through 9, including the details of which ion channels and pumps are present, what the term voltage gated means, and at what voltage each of the channels opens. To finish our introduction to the action potential, you'll also discuss the term membrane potential, which you'll do on slide number 10. You'll also discuss how the membrane potential is generated on slides 11 and 12, as well as how it appears on a graph on slide 13. The next section of the discussion describes the steps of the action potential by examining the movement of sodium and potassium ions into and out of the axon. Each of these slides is animated with questions so you can ask your students to predict what would happen and then show them. Slide 14 allows you to describe what happens in the axon after a neuron is stimulated and a positive charge reaches the axon's first set of sodium channels. They open if threshold is reached and sodium ions rush into the axon. Slide 15 demonstrates how this flow of ions appears on a graph and introduces a key vocab term, depolarization. Slide 16 allows you to describe what happens in the axon after depolarization. Sodium channels close, the potassium channels open, and potassium ions rush out of the axon. Slide 17 allows you to demonstrate how this flow of ions appears on a graph and introduces another key vocab term, hyperpolarization. Slide 18 allows you to describe what happens in the axon after hyperpolarization. The potassium channels close, and the sodium-potassium pump reestablishes the axon resting membrane potential by pumping sodium ions out of the cell and potassium ions into the cell. Slide 19 demonstrates how this movement of ions would appear on a graph. The key points of the activity are to demonstrate the creation and conduction of the action potential down the axon and review the forces that govern that process, including the impermeability of the cell membrane, sodium-potassium ion channels, and diffusion. To review the flow of sodium and potassium ions during the stages of the action potential, including at rest, to getting to threshold, depolarization and hyperpolarization, and then again returning to rest. We'll get to these key points by modeling the action potential. In the activity, you'll have students work in small groups and give each of them an action potential model setup, which has the supplies for them to simulate the action potential, using dried beans to represent ions and toothpicks or pipe cleaners to represent the ion channels. You'll also give each student an activity worksheet to help guide them through the activity and help them focus on the most important details of the process. The worksheet has students take notes on those key points, which are where are sodium and potassium ions at highest concentration when the axon is at rest? Which direction do sodium ions move after the first sodium channel reaches threshold and opens, and why? How does the movement of sodium ions change the charge inside the axon? Which direction do the potassium ions move after the first potassium channel opens, and why? And how does the movement of potassium ions change the charge inside the axon? After students have had time to model the action potential themselves, you'll come together as a class and review the entire process using slide 21. This slide is heavily animated, so you'll walk through each step of the action potential with your students, as well as highlight how the action potential occurs several different times down the length of the axon. Again, if you need to review these steps yourself, please see the teacher primer or teacher manual. Also, an important note here, this lesson does not discuss how the action potential is converted into a chemical signal at the synapse. That process will be discussed in Unit 3. The key point of the wrap-up is to think about the effect of disrupting the action potential, and we'll get there by examining how Novocaine works. In the wrap-up, you'll revisit the question, how does Novocaine work? Slide 22 shows students that Novocaine inhibits the voltage-gated sodium channels and asks students what would happen if these channels can't open. If the sodium channels can't open, then we can't send an action potential and you won't feel any pain. The key points of the homework are to review the creation and conduction of the action potential down the axon, as well as the flow of sodium-potassium ions during the stages of the action potential. We'll get to these key points by completing the activity worksheets. The final section of the activity worksheet has students describe what is happening at each stage of the action potential to generate the graph we examined in class. If students need help, they can read Lesson 2.2 in the workbook to review the steps of the action potential. Students are often challenged by the content in this lesson, so it is important that you understand the concepts well so that you can guide your students. You can find more in the teacher primer, the teacher manual, and the student workbook. One of the most common questions teachers get is, where does the positive charge come from after the neuron was stimulated? This is referring back to slide number 14. It's an awesome question because it shows your students are really thinking about how all of this comes together, and we did skip over that part. The answer to this question lies in the fact that neurons have different types of signals: electrical signals down the axon and chemical signals across the synapse. The positive charge that starts the action potential results from synaptic signaling and will be the focus of Unit 3. After teaching the action potential, a question I like to ask my students is, what would happen if the sodium channels in your pain-sensitive neurons never reached threshold? This question mimics the Novocaine question by assessing student understanding of the entire process and has the added benefit of acting as a bridge to an actual disorder and patient video. The answer here is that you'd never feel any pain, because if your sodium channels don't open, you can't fire an action potential. So regardless of how much pain you're in, a message would never get sent to the brain to tell you that you are in pain. And this does happen to people with congenital analgesia. One of these patients was profiled in a documentary called A Life Without Pain. We'll discuss this patient and the condition in more detail in lesson 2.5, but it's nice to introduce here and to show a clip of one of the many videos of it on YouTube if time allows. At the end of this lesson, students should be able to describe the process by which our neurons generate and conduct an action potential down the length of the axon. They should also be able to predict the consequences of disrupting this process. An understanding of the action potential is important because it describes why myelination is important, which we'll discuss in the next lesson, and it explains the causes of some of the disorders we'll discuss in lesson 2.5. Additionally, the action potential is just one type of neuronal signal. It leads to synaptic signaling, which is the focus of Unit 3. And don't forget, if you have any questions, concerns, or feedback, to let us know. You can contact any of the CTSE team members, and we'd be more than happy to help you.